Hello, everyone. My name is Gert Lawler, and on behalf of the Foundation for Conservation of Atlantic Salmon, I would like to welcome you to today's webinar. Today's webinar will be presented in English. However, we have AI translation available. If you wish to use the translation service, please navigate to the chat where you will find instructions on how to connect. For today's webinar, we're very pleased to be hosting Craig Purchase, Dr. Craig Purchase. Craig has a Bachelor of Science honors and Master of Science from Memorial University in Newfoundland, and a Doctor of Philosophy from the University of Toronto. Following his education, he worked as a postdoctoral fellow at Dalhousie University, a lecturer at St. Mary's University, a research scientist at the Bedford Institute of Oceanography, and an NSERC visiting fellow at the Northwest Atlantic Fisheries Center. Since 2008, he has been a professor of biology at Memorial University and is cross-appointed to the Department of Ocean Sciences and the School of Fisheries. He is also a supervisor in the Cognitive and Behavioral Ecology graduate program and the Environmental Science graduate program. He has held several other appointments, most recently serving as president of the Canadian Aquatic Resources section of the American Fisheries Society. Today, Craig will be speaking on reproductive quality of virgin versus repeat spawning Atlantic salmon, Salmo salar, and how different diets may lead to altered hybridization threats from invasive brown trout, Salmo truta. After this afternoon's presentation, we'll be opening the floor for questions and answers. And now I will turn the webinar over to Craig. Welcome, Craig.
Thanks, Gert, and thanks, folks, for those of you who have logged on. Gert already read my title, so I'll move right in here. I'm battling the plague today, but I think my voice is going to hold up. So first of all, I'd like to acknowledge the many groups and people and funding that enable this type of work to happen. First and foremost, my current and former research team at Memorial University, also Fisheries and Oceans Canada, where I've had great collaborators and also logistics help and permitting help on many fronts. IRMA, a stewardship group based in Grand Falls, Newfoundland, where I've had many years of collaborative work and a large project that's ongoing now that I'm not going to talk about, but the work I am going to talk about today would have been impossible without the help of IRMA. The ideas that I'm presenting here have been formed with collaborators that are based in various countries around the world, and this work has been funded by numerous fronts, including several grants from the Foundation. I'm going to start in the big picture here today and paint a picture about how selection happens through the life cycle of animals. In all sexual eukaryotes, that would be all animals and plants and other things, this process is basically the same. So this looks like a complicated cartoon, but I'm going to go through this one step at a time, and it's important for then how we connect the dots later. So in a generation begins with the fertilization of an egg and the formation of a zygote, which then develops through time and through the juvenile state to an adult. And these individuals have a copy of the genome from their mother and their father, and so therefore they're called diploid. And this process is the same across all animals. And of course, in different species, it might take a few weeks to reach adulthood. In other species, it might take many decades, but more or less the nuts and bolts of this are the same. Animals undergo gametic meiosis and produce gametes, and the little gamete is male and call them sperm, and the big gamete is an egg, which is female, and some of those fertilize and then produce the next generation. And these gametes are haploid. They only have half of the genetic material as what the diploid phase does. And in many animals, this diploid phase takes a long time. So for example, in an Atlantic salmon from Newfoundland, this would on average take about 5 years. But this haploid phase, you know, the meaningful part of this in an Atlantic salmon may only last about 10 seconds. But we have to think about how selection acts on both phases of this biphasic life cycle. So these are some simplistic definitions and illustrations of this. So with the formation of a zygote and a new generation, these individuals are then exposed to post-zygotic natural selection. So we can think of this as what Darwin described, or Darwinian natural selection. And in its simplest form, I call this basically a zygote to adult survival and the ability to produce and release gametes. So, or we can think of this as who lives to mate. And of those that live to mate, they don't all mate with the same opportunities, and this because selection is occurring in other forms. And we might think of this as post-zygotic but pre-mating sexual selection. So of those that live to mate, who gets to mate? And this is a function of competition within a sex, such as male-male combat or access to territories and access to females, and also between the sexes, such as mate choice. And in most animal species, the females are the choosier sex. Once mating occurs, this is not the end of it either. So once gametes are released, selection happens on those. And it's a little bit difficult to visualize natural selection on sperm and eggs. It's much easier in certain types of plants where this process takes a long time. But we can think of, for example, salmon gametes as post-mating pre-zygotic natural selection. And we can think of this as egg survival and fertilization capability, and sperm survival and fertilization capability. So for example, if the adult produced gametes that, wouldn't function, selection would remove, this process pretty quickly. Of those functioning gametes, not all of them fertilize to the same extent. And because the last form of selection that we occur here is post-mating pre-zygotic sexual selection. So on the functioning gametes, we see intrasex selection. So for example, sperm competition, and this is where the ejaculates from more than one male compete to fertilize a given set of eggs. And this is ubiquitous basically across almost all animals, but we didn't have any idea that this even existed until the early 1970s. And then when sperm competition is happening, there's also intersex selection, for example, where females can bias the outcome of sperm competition towards certain males. And we'll come back to this later. So my research approach and the types of stuff that I do is varied. And so I do what many people on this call might often do, and that is measure or monitor things from wild fish. So this could be things like juvenile densities or juvenile growth rates, pure ecology type stuff. But I also do a lot of measurements on, for example, adult fish status or their quality, things like age and their shape and their size or some measure of their health status. But I do a lot of work also on gamete quality produced by adults. But in all these cases, we're basically measuring stuff that naturally exists in the wild. The other type of research I do is experimental manipulations in the lab. And here we're manipulating either the precise conditions of the fertilization microenvironment or the incubation conditions that embryos experience. And that we do this by doing split ejaculate experiments or split brood experiments. And as a result, we monitor sperm or offspring performance. And some of the work, including some of the things I'll talk about today as an example, kind of connects these two, where perhaps looking at adults of different health status and then how that interacts to influence, for example, the performance of their sperm and sperm competition in different contexts. So a lot of the work I do is with salmonids and particularly Atlantic salmon, and this is an Atlantic salmon talk. And so we can start here by looking at this pretty diagram of the salmon life cycle. And this is a very simplified version of the Atlantic salmon life cycle. Atlantic salmon are basically the most diverse species on the planet in terms of the different types of life histories that they have. This illustration is almost so simplified it becomes useless, but it does become, therefore, a good teaching exercise. So Atlantic salmon are freshwater fish. They are tethered that they must reproduce in fresh water, and they may or may not go to sea. They reproduce in the fall, and the eggs are generally buried in gravel and incubate through the winter. And then the young ones emerge and start feeding in the spring. And there's lots of jargon when it comes to Atlantic salmon terminology, but basically in their first summer, people call them fry, and then later summers, they call them par. And some of those, and the proportion varies in different places, but the general idea for anadromous salmon is that in the spring, once they reach a certain age or size, they undergo some physiological changes to be better adapted to salt water, and they're called smolts. And they go out to sea for various amounts of time where there's a lot more opportunities for feeding. However, there's a lot more predators too, so it becomes a bit of a tug of war about the pros and cons of doing this. But they feed much more effectively in the ocean and grow a lot quicker. And after some amount of time in the sea, they come back to fresh water, generally close to their natal river or to their natal river, and then reproduce in the fall. But this general idea of the salmon life cycle hides a lot of diversity. So for example, there are resident forms that never ever go to the ocean. We often call these Wananish, which Newfoundlanders interestingly pronounce Winnanish. And these are males and females. It's typically associated with rearing in lakes, although there's interesting exceptions to that. And there's also alternative reproductive tactics where, and this would be in males, where they mature precocially. So if we go back to this illustration, this is meant to illustrate a spawning adult female. This is a spawning adult, large, dominant phenotype male. But this is also an adult male down here, this little guy. This matured basically as the morphology of a par, but he's still an adult male. For anadromous fish, these are fish that are going to the ocean. We get variable age at the seaward migration. So for example, it could be smolts from ages 2 to 7 in our part of the world. The modal age in Newfoundland is 4, but in the Maritimes, it's younger than that. Once they go to sea, in their first trip, they can stay at sea for various amounts of time. So some individuals, and these are typically males, only go out for a few months, and by the end of the summer, they're back in the river again. And these are called post-smolts, and these are first-time virgin spawners. Other individuals go out to sea in the spring, and they stay out a whole year, basically, maybe a little bit more, through one winter, so one sea winter. And Atlantic salmon jargon calls these grills, and this is by far the most common type of anadromous spawner in Newfoundland, but not in some other places. Other individuals, when they go to sea, they stay at sea for 2 or more winters, and all of these are classified as multi-sea winter. The longer they stay out to sea, the bigger they get, but the higher they risk that something is going to kill them before they come back. But all 3 of these types are virgin or maiden spawners from anadromous fish. The Atlantic salmon, unlike Pacific salmon, are iteroparous. They don't all die after reproduction. Most of them only reproduce once, but many reproduce more than once. And we can classify those in different forms. So consecutive repeat spawners are fish that, for example, spawned last year and will spawn again this year. So they don't skip a year. But alternative repeat spawners, for example, might have spawned in 2023, but not 2024, but will spawn again in 2025. And these things interact. So you could have a fish, for example, that spawns 2 years in a row and then skips a year. And all of this basically magnifies up through the page here. All these things interact. So at any given time in a population, the background of the adults that are spawning is very diverse. This enables one of the resiliency of the species. We've been working for quite a few years now on some aspects of two of these spawner types. And so I'm going to go over the differences here and what these are. So virgin or maiden grills are, again, by far the predominant type of anadromous spawning adult salmon in Newfoundland. These leave rivers as juveniles in the spring. They migrate relatively long distances compared to the next group. They stay at sea about 14 months. They feed substantially on invertebrates, and there's a little asterisk here because this may be important to our story. The following spring, they cease feeding and they enter rivers in early summer, usually, and then they spawn in November. And again, this is by far the most predominant spawning type. Consecutive repeat spawning grills are the fish that did that the year before, and they survived their spawning in November. They remain in fresh water for another 6 months, so they haven't fed at all for about a year. They migrate back to sea in May, usually a little bit earlier than the smolts, and they're called kelts at that time. Interestingly, Newfoundlanders call them slinks. We know from other people that they stay coastal generally, and those that spawn consecutively, they stay coastal, they don't go very far in the ocean, and they return to the river after only a few months. So by late summer, they're back in the river again. And in that time, they fattened up. So they went out to sea in May as a very skinny, poor condition fish. They fattened up and came back, but they gained very little in length, only about 2 centimeters on average. When they were at sea and fattened up, they fed largely on fish, and I think capelin are important here. I think the lucky fish that do spawn consecutively, most of them hit a school of capelin and basically pig out and then turn around and come back. And they spawn in consecutive November, so they haven't skipped a year. So some of those things we know from the work of other people. So for example, work from Ian Fleming's group and collaborations with the DFO showing that particularly on the northeast coast of Newfoundland where this was done, these consecutive spawners don't go very far when they're out at sea. They don't stay out very long, and they have a diet that's different than the first time spawners. So from other people, we know that these two types of spawning fish have different migrations and diets. And from different people, we know that the diet impacts carotenoids, which are the red, orange, yellow pigments in animals. Animals can only get these things from their diet, and that these carotenoids are important. Some observations that I had before this work started. For example, I knew there was variation among salmon in their flesh color. So for example, a lifelong time of being an angler, seeing lots of people catch salmon and eat them, sometimes you get a salmon, you cut it open and it's really paled flesh, and most anglers are really disappointed when they see that. I also knew from my work with salmon reproduction that among different females, there was lots of variation in egg color. And this got me thinking about whether there were consistent reasons for this. So the hypotheses that started some of this work was that the repeat spawners, and particularly consecutive repeats, have lower carotenoids in their flesh than virgin spawners due to having a predominantly fish-based diet for repeats, but having a much more substantial invertebrate-based diet for the virgin spawners. And as a consequence of this, at spawning time, thinking that repeat spawning males are duller in color and perhaps have poorer quality sperm, and females have poorer quality eggs as repeats versus virgin spawners. So to get data on this, started doing work on the Exploits River. And so the Exploits River drain is the largest watershed in Newfoundland, and it enters the ocean on the northeast coast in Botwood, which is coincidentally where I grew up. And IRMA is based on the Exploits River at Grand Falls, and this is their interpretation center. I think you can see my cursor. I hope you can as I'm pointing the things here. And this is the lower salmon ladder that goes around Grand Falls. So the fish that we got for this work came out of this salmon ladder. So they're all wild fish taken right from the ladder. And the data I'm going to show you here came from males that were collected in 2016 and 2018. And this was for a master's project for Madison Phillip. And the female salmon were collected over 3 years for a PhD project of Brittany Palm. And the scales were taken from these fish when they were sampled for their gametes and sent to the DFO Newfoundland, who kindly had their normal technicians who aged and interpret for their monitoring programs. They interpreted these fish for us, and about 10 to 15% of the sampled spawning fish were repeat spawners, and the vast majority of those are consecutive repeat spawners. So if we go back to our illustration of the anadromous salmon life cycle, we have fish going to sea feed, and when they went out as smolts, they're fairly small and they can't feed on big things. Maybe they can feed on lots of little brightly colored invertebrates, crustaceans. Or when they go out as a kelt and they're much bigger, they can feed on that, but they can also feed on bigger things like maybe these dulled colored fish, which maybe or maybe not capelin in this illustration that I didn't make, but you can see from my story here that this illustration shows a varied diet, and that varies a lot in carotenoids. And salmon take the energy they get from their food and the pigments and they put it in their flesh. And so when they come back to spawn, the males take this stored pigment from their flesh and they put it on the outside of their bodies. So Pacific salmon, as an example, think of a sockeye salmon that is all bright red. But Atlantic salmon do this too, and they become bright red and showy. And the general idea is that they'll achieve more mates if they're more brightly colored, either because of aggressive responses with other males, or maybe females like more colorful males. But the general idea is that the redder they are, the better. So again, the hypothesis here for males is that repeat spawners have lower stored carotenoids in their flesh than virgins because of different diets in the months prior to spawning. And that as a result of that, repeat males have duller coloration than virgins during spawning. And repeat males may also have poor quality semen, and there's reasons to expect this based on literature with carotenoids. So here is a male Atlantic salmon from the Exploits River in his fall tartan, and you can see that how nicely colored he is, but they all don't look like this. Over here we see on the left, these are some pictures from virgin spawners, and on the right, pictures from repeat spawners. There's only 4 animals of each type shown here, and pictures have been picked, but you can clearly see there's a difference in color. And we quantified this in ways I'm not going to get into. But if we, on the y-axis here, we have the proportion of the body that has red pigment, and we have the virgins, fair amount of variability amongst them, and then the repeats. And this is the most red fish that we sampled and the most unred or the most dull fish on the right. So this is a consecutive repeat spawner. This is a virgin spawner. And you can see the data clearly shows that the consecutive repeat males are on average much duller in color. But what about their semen? In my lab, we do a lot of work on assessing various attributes of fish semen, and exactly how we do that, I'm not really going to get into here for time. But if we look at metrics that might be indicative of the fertilization ability of the semen. There are 3 metrics on this slide. In each case, the virgins are plotted on the left and the repeats are plotted on the right. So the first one here is the semen density. So this is the proportion of the semen sample that is actually sperm cells. And although the box plot might be a little lower here, because of the error around this, there's no difference in these 2 groups. So there's at least not much difference between the virgins and the repeats in the density of their semen. There's also no real difference in the proportion, there's an ambulance going by behind me, proportion of their sperm that actually swim or are motile. And of the sperm that swim, there's no difference in how fast they swim. So at least no dramatic difference in what I would conclude is the ability of their semen to fertilize eggs between the virgins and the consecutive repeats. Now for females, again, they go to sea and they eat different things. And we know that the first time spawners, which over much of their time at sea, they were small individuals, and the consecutive repeat spawners, which when they went out prior to spawning, they were already big. We know that they eat different things. I think that they have different stored carotenoids in their flesh, repeat spawners with dull flesh. And the females take these pigments and put them into their eggs. So why salmon have red, orange eggs is a different question, but this is how they get red, orange eggs. The females take these pigments out of their flesh and they move them into their eggs. And the eggs vary in color. And first, when I was looking at these, the head scratching was going on. Well, is there a pattern here in the virgins and the consecutive repeats? And I can now tell you without a doubt that there is. And on the left here, these are eggs from a consecutive repeat spawning salmon, and the ones on the right are from a virgin grills. And you can quantify the color in different ways. I won't get into different ways that we did this, but for example, this is a chicken egg yolk fan, and then the beaker, same volume of eggs from the, on the right is a consecutive repeat spawner, on the left is a virgin spawning grills. And this is very consistent. I've only looked at fish from the Exploits River, but in the exploit system anyway, there's a very consistent difference in the color of the eggs. Not surprisingly, we would expect to see differences in the egg chemistry, and we've quantified maybe 40 parameters related to egg chemistry, but this plot just shows one. This is a certain type of lipid. Excuse me. On the left here, we have virgin spawning grills from 2014 and consecutive repeat spawners from 2014, and then from 2015, the same thing. And in this particular chemical, you can see that there's very dramatic differences in the egg chemistry between the 2 spawner types. But what about the ovarian fluid? So in animals, female reproductive fluids play an important role in fertilization, and in fish, we typically call these ovarian fluid. And this is the viscous liquid that is released with the eggs. Basically, all fish have this, but in salmonids, they have a relatively large amount of it. We know from several decades anyway of work on people working on aquaculture and cryopreservation and all kinds of different things, that the chemistry of that ovarian fluid really matters in terms of fertilization dynamics. And a little side project we did with the fish from the Exploits River, we show that the physics of it matters too. So this was a paper we published a few years ago based on the Exploits River salmon, where we show that the non-Newtonian nature of ovarian fluid seems to be well lined up to explain many aspects of sperm-egg interactions in fertilization. Ovarian fluid typically helps sperm and fertilization dynamics. And this is another little side project that we published related from gametes that came from the Exploits River system. The 3 panels here on the left, these are lake trout from Ontario, Panel C on the right is brown trout from near St. John's, but the middle panel from B are salmon from the exploits river. What this diagram is the swimming velocity of sperm in water, which is the blue line, or in ovarian fluid, which is the red-orange lines. And the two lines are just whether the ovarian fluid was frozen first or not. But the point here is that, just illustrates that in the presence of ovarian fluid, the sperm swim faster than if they had just been in water. And again, this is a little side project from our story. So to summarize a bit with these virgin and repeats, we know that the virgin and consecutive repeats have different diets prior to spawning, and we think they have different stored carotenoids because of that. Repeat males do have duller sexual coloration. This likely leads to less mating opportunities. Certainly wouldn't expect it to lead to more mating opportunities. But they seem to maintain their sperm quality, at least in its fertilization capacity, which means if they, were present for spawning, I think they would fertilize eggs fine. Repeat females do have duller eggs that do differ in chemistry, and I think are of poorer quality. Don't have as good data on that part, but some. And the females are making these eggs from the energy reserves that they gathered in their diets, and so they're probably, making the ovarian fluid that's released with the eggs, at the same time and with the same resources. So if the eggs between virgin spawners and repeat spawners differ in chemistry, for example, I would think, although I don't have data on this, that the ovarian fluid varies in chemistry as well. And then this moves us into talking about what happens after the gametes are released. So if we go back to our cartoon, I was talking about, the characteristics of the fish and of their gametes, but then what happens in the haploid phase before fertilization. And we're going to talk in particular here about cryptic female choice and how it changes the outcome of sperm competition. So female reproductive fluids have been repeatedly shown to impact the outcome of sperm competition. In external fertilizing animals like salmon, this is really the only way that females can change the outcome of sperm competition. And if, for example, the female reproductive fluid changes sperm performance and changes it for different males in different ways, then this is a mechanism that it can bias paternity under sperm competition towards certain males. And if we think about this as intraspecifically, so among males of the same species, this could be a mechanism where females can bias paternity towards certain individuals, so, certain blokes, one versus the other. But we can also think about this interspecifically, and I personally think this is far more important. And when this occurs, this is called conspecific sperm preference. So when there are sperm in competition from multiple species of males, conspecific sperm preference allows the female to bias the paternity towards her own species. And that reduces the hybridization of a female's eggs when there's sperm competition between conspecific sperm to her and heterospecific male to her. And that leads us into our topic on hybridization. So what is that? Hybridization, is a result, but also a process. If we think about a female horse and a male donkey, these are closely related species that can hybridize. And in this case, they produce a mule, which through human history has been exploited and purposely created for many things. But interestingly, mules are sterile. So it's an outcome, this is a classic example of a hybrid between related species. Hybrids are a real head scratcher because we have to think about what causes a hybrid and how can a hybrid be reproduced. And there are many things that have to happen in order to get a hybrid individual. And bottlenecks or problems anywhere along this chain of things will prevent the presence of a hybrid. So I'm going to go through this. So we have to have two species that are reproductively active in the same place at the same time. So for example, this could be a salmon and a human and a sheep. There has to be heterospecific mating, so a breakdown in pre-mating sexual selection barriers. So, you know, theoretically, this could be a human sperm with a salmon egg, it could be human sperm and a sheep egg. There has to be gamete compatibility. Human sperm will not fertilize a salmon egg. I'm not sure if it would fertilize a sheep egg or not, but a donkey sperm will fertilize a horse egg. If the gametes are compatible, there has to be a breakdown of these sexual selection barriers to reduce hybrid fertilizations. And so, you know, the conspecific sperm preference falls in this area. So anywhere in here where there's a problem, like an absolute cutoff or a real bottleneck, these are prezygotic, so before fertilization, barriers to prevent or reduce hybridization. And so for a female whose eggs might be lost to hybridization, you know, having mechanisms to along here would be highly adaptive. Once eggs are fertilized by a heterospecific male, and this could be a sheep fertilized by a human sperm, I don't know, but they certainly don't hatch. So there's postzygotic survival that is needed for development to occur for long enough for a hybrid individual to be present that someone can go and find or catch and identify it as a hybrid. So if all fertilized eggs like don't hatch, or they hatch and they die pretty quick, or the juveniles die relatively quick, then you'll never ever see a hybrid. And if you never ever see a hybrid, you may incorrectly conclude that none of this process is actually going on. And so because of this, you know, for example, if 1% of juveniles in a population are hybrids, this really tells us nothing about how common hybrid matings are or hybrid fertilizations are. It only tells us that some make it through. It tells us really nothing about how much the process is in the beginning. And so in external fertilizers, females can be as picky as they want. The female can spend a lot of time sizing up different males, and she can be really choosy at picking Mr. Right. But when she goes to spawn, many males can release sperm. She has no control over being able to prevent them from releasing sperm. And when they do, all but one were not the female's chosen mate. That type of thing is very common in salmonids. Some of those males that release sperm when she picked Mr. Right might have been heterospecific to the female. And if they were very distant related species, perhaps the gametes are not compatible. But if they're a closely related species, perhaps they can fertilize the eggs. When this happens, cryptic female choice is the last line of defense to prevent her eggs from being hybridized. And if they're hybridized and they have poor survival, then, you know, they're totally lost. So she's lost really all that energy that she put into that. So the question becomes, well, how good is this mechanism? And so if we go back to our cartoon again, we have fish that have become adults in fresh water, and they're going to spawn. And our diagram here is a real oversimplification. We have a female here shown spawning with one bright, showy male, but in reality, there can be all kinds of fish that look like him or similar to him in and around the mix. And even if she decides to spawn with this guy, these other fish will come in and throw sperm into the mix very quickly. We also have these little adult males, these precocial par, and in this diagram, there's an image of one of these, but usually there's many of these in and amongst this process. Some work from our friend Laura Weir, who's based in St. Mary's in Halifax now, from time ago, showed that in salmon nests, she found up to 16 different fathers contributing to the paternity of a given female's eggs. And so my point then becomes some of these different males might be not her species. It could be from a closely related species. And so she might have picked the spawn with her own species, but she can't prevent the other males from releasing sperm, but cryptic female choice might be able to allow her to bias the paternity towards the sperm of the right male. So if we think about hybridization in salmonids, which is common, we did a little review on this a few years ago, and this is a table coming. So this is Atlantic salmon. The sister species is brown trout, Salmo truta, and Salvelinus fontinalis is the brook trout, which is a char. I've called it a char on here to make the wording simpler for a trout. So this little table below, in each case, the female is on the left, the male is on the right. And so if we think about Atlantic salmon and brown trout, and then brown trout and Atlantic salmon, etc. So we see hybrid individuals of Atlantic salmon and brown trout in nature. Talk about this in a minute. We don't generally see or never see hybrids between salmon and brook trout, brook char. And the question is why? And the point of this table is it's not because of fertilization. In vitro, in the lab setting, the gametes are fully compatible. You can produce a hybrid between a salmon and a brook char. It has to do with what happens after. And the hatch success is pretty good between hybridizing Atlantic salmon and brown trout. It's not very good between hybridization and the other groups of species. This paper was published in 2013. I saw it about 6 months before it was published, and it has formulated much of my thoughts in the subsequent 12, 13 years. Sarah Yates is the lead author. It was published in Evolution. It is a pretty amazing study done in Europe on brown trout and Atlantic salmon. And in Western Europe, brown trout and Atlantic salmon co-evolved. They occur in many of the same rivers. They spawn often in similar places. They have spawning seasons that overlap somewhat. So there's a constant threat of hybridization between the two. What these figures here show, for example, is that the gametes are fully compatible. So if we take salmon eggs here on the left, and we look at these fertilization trials in the presence of one male, so if we take salmon eggs and give them conspecific salmon sperm, or in the dark bar, we give them heterospecific trout sperm, there was no difference in the fertilization success. Similarly, over on the right, if we take trout eggs and we give them conspecific trout sperm or heterospecific salmon sperm, there's no difference in the fertilization success. So in the absence of sperm competition, the eggs of both species are fully compatible with the sperm of the other species. But if you have a sperm competition scenario, like may often exist in nature, we get a very different result. So under sperm competition for salmon eggs, most of the eggs are fertilized by conspecific salmon sperm, but some are hybridized by the heterospecific sperm of trout. And in trout eggs, under sperm competition between salmon and trout, most of the trout eggs are fertilized by conspecific trout sperm, but some are hybridized by the heterospecific salmon sperm. And this shows that the females of each species exert conspecific sperm preference. It's not perfect. They can't eliminate hybridization completely, but they can bias fertilizations towards their own species, even though the eggs are fully compatible with the sperm of the sister species. What further was really cool about that study was some window on how this happens. And this is a complicated figure, but it shows that the ovarian fluid that is released with the eggs clearly influences the outcome of paternity under sperm competition. But exactly what it's doing or how it's doing that is not as clear. So here we have salmon eggs and salmon ovarian fluid. That would be the natural situation. And we have salmon sperm here. So salmon eggs, if there's salmon ovarian fluid present, the natural situation, salmon sperm achieve a higher fertilization success than if we took the ovarian fluid away from the salmon eggs and we replaced it with trout ovarian fluid. And if we do that, the percent of the eggs that are fertilized by salmon sperm drop. Similarly, for example, if we take trout eggs in the presence of trout ovarian fluid, the natural situation, salmon sperm fertilize less of the trout eggs than if we took the trout ovarian fluid away and replaced it by salmon ovarian fluid. That helps these salmon sperm and they achieve more fertilizations. And the same pattern occurs here on the other side. So this clearly shows that the ovarian fluid is doing something, and the presence of the ovarian fluid affects the outcome of sperm competition and changes it, which species actually wins. So again, for hybridizing Salmo, for Salmo salar, Atlantic salmon, and Salmo truta, in their native Europe where they co-evolved and reproduced together, hybrid fertilizations do exist. You can find hybrid juveniles in nature, but these hybrid fertilizations appear to be reduced by cryptic female choice, and that ovarian fluid seems to be important in mediating that. So the ovarian fluid affects the hybridization when there's sperm competition. That work, though, that was published in 2013 in the journal Evolution, was done with one population of each species, and they were from Norway. And one of the things that I have been pondering ever since is that whether the strength of that mechanism is dependent on reinforcement. So in other words, if the two species are constantly in contact, then the importance of this would be high. But if the two species are virtually never in contact, well, then they don't need this. And maybe if that's the case, the strength of it is lost through time. So if that was the case, then historical allopatric and sympatric relationships between the populations of the species is likely important for how strong this mechanism is. And so then this gets back to brown trout and where they don't belong. Brown trout, which are native to Eurasia, were widely introduced around the globe over 100 years ago, and basically anywhere where appropriate freshwater temperatures exist. And they're now considered one of the world's 100 worst invasive species, and not 100 worst invasive fish, but of any species. And usually the context here is with ecological things and competition with other species. And the big conservation concern is how they affect galaxid fishes in the southern hemisphere, where there are no native salmonids at all, and with Oncorhynchus Pacific salmon and trout in Western North America. But also less studied would be with, for example, brook trout or Atlantic salmon in Eastern North America. What is generally not studied, though, are the evolutionary impacts of these invasive brown trout. And we can think of this in two contexts. One, we think about in their native range, we would have locally adapted brown trout populations, and the introduced brown trout may be hybridizing among populations of brown trout. So for example, populations from the Caspian Sea, their local adaptation may be being evolved by brown trout that were introduced from Western Europe. But we can also think about brown trout hybridization with other species. I mean, they can't hybridize a galaxid or a carp or a sturgeon. They have to hybridize species that are closely related enough that they can hybridize. And this is most relevant in Eastern North America, of course, where we are based. The introduction of brown trout is actually pretty well documented. And so, for example, here are some images of ground zero, maybe. So this is from the Howatton Hatchery, which is near Stirling in Scotland, and images of a book, for example, on how this was actually done. And what they would do is they would fertilize the gametes, and they realized that if you wait a few weeks or months, depending on the temperature, for them to develop to the eyed stage, then they get quite a bit more robust. They're not as sensitive to vibrations, and they would package them up in wooden crates with ice that would then melt and drip on them, and they would put them on a ship, and they would send them around the world, including to St. John's, Newfoundland. So they were brought to St. John's, Newfoundland, and to other places. New York was around the same time. And we know, for example, that in St. John's, at the city's water supply at Windsor Lake, is the first confirmed established population of brown trouts in North America. And they were introduced for other parts of eastern Newfoundland on the Avalon Peninsula, and they are spreading west. And we've recently published an update on this, on the spread rate and some predictions. And some interesting things come out of that. They are spreading west, but in terms of the global picture, it has been very slow. So it's actually the spread rate of brown trout in Newfoundland is the slowest documented spread rate of anywhere that they've been introduced on the planet. And, you know, it becomes a head scratcher as to why. So perhaps the island's low productivity in fresh water, we have a very oligotrophic environment, juvenile growth is slow, therefore, you know, the population growth rate would be low. It's possible it's related to a competition with native salmonids. So in all the rivers they could move into Newfoundland, there are brook trout. Almost all the rivers, there are Atlantic salmon. So, you know, there's some biological resistance from closely related species that would, you know, keep them from really establishing themselves. And a totally unrecognized thing, I think, is with hybridization. So, for example, if female trout, as they spread west, if their eggs are being hybridized by brook trout or Atlantic salmon, then this would limit the trout's ability to colonize new environments. This particular work that we published last year had some predictions. And so, for example, based on the past rate of spread, it is predicted that brown trout will reach Terra Nova National Park by the year 2054. So I've had a bunch of different brown trout hybridization questions in my mind, some of which we've started work on, some of which we haven't. So, for example, how much of an impact does brown trout hybridization have on Newfoundland's native Atlantic salmon and brook char? They hybridize with both. The hybrid between a brook char and a brown trout is called a tiger trout. And so this would come from male trout hybridizing female salmon or char eggs, and for example, reducing the productivity of a salmon population. I personally think this might be a big problem in particular areas. The other flip side of that is, though, is hybridization an explanation for the slow spread of brown trout in Newfoundland? It is the slowest of anywhere where browns have been introduced. It is the most chances for hybridization where anywhere where the browns have been introduced. And this would come from male salmon or male brook char hybridizing female trout eggs and reducing the productivity of the invasive trout to colonize new environments. The last bit of work I'm going to talk about here is the master's project of Connor Handley. So a little bit of background here and context again. So North America Atlantic salmon and European Atlantic salmon are the same species, but they're quite different from each other. Genetic work has indicated that the salmon in North America have been isolated from those in Europe for at least 600,000 years, and they actually have different numbers of chromosomes. And with this context, we are repeating the work that Yates did with Norwegian fish with fish from Newfoundland, with Atlantic salmon from Newfoundland, and with brown trout from Newfoundland. And we are looking at testing the hypothesis that the hybridization rate of Newfoundland salmon eggs during sperm competition is higher than what is reported in Europe, but less difference in the trout eggs from Newfoundland and Europe. And the reason being is that if there's sperm competition between a male salmon and a male trout, our female salmon in Newfoundland have evolved in the absence of trout for at least 600,000 years, maybe much longer. They never needed the mechanism to exclude trout sperm. Perhaps conspecific sperm preference has been lost compared to the European cousins. But the female brown trout that are in Newfoundland have only been isolated from salmon from Europe for less than 200 years. And so they probably have the same mechanisms that their long lost cousins back in Scotland had. A follow-up from this and connecting our dots from earlier on is testing the hypothesis that due to poor egg quality, which I think leads to poor ovarian fluid quality, the hybridization rate during sperm competition is higher in consecutive repeat spawning salmon than in virgin spawning salmon. So some data here. This was a paper we published last year and a bit of a review, but this is the only data figure in it. This is with Newfoundland fish and shows that, like in Europe, the gametes are fully compatible. This is actually the hatch success. Each line is a female and the two dots are the type of male. So here we have a female trout whose eggs have a trout father, or then the sibling eggs, the split batch, where the father was a salmon. So that's a trout hybrid. Over here, we have a salmon female who's been, half of her eggs are fertilized by a salmon male, and half of her eggs on the other side of the line are fertilized by a trout male, creating a hybrid. What you can see is there's generally a decline in these lines, but it's not very pronounced. So when we mix these gametes together, the hatch success is poorer for the hybrids, but not by very much. And the fertilization success undoubtedly is basically the same. So like in Europe, the gametes are fully compatible. So when there's no sperm competition and you just give the eggs one species of sperm, it really doesn't matter which species of sperm it is, the eggs fertilize and develop. So this is a picture of a spawning brown trout. These are the ones we've been working on near our campus in St. John's. And we get the gametes from spawning tributaries that are only about 10 minutes drive from the campus. And through coordinated efforts, we collect the gametes from the brown trout at exactly the same time as IRMA staff collect gametes from Atlantic salmon on the Exploits River. And so literally we are on the phone or texting like, okay, we're sampling the fish now, go ahead and collect the gametes. And in St. John's with the trout, we package them up in a certain way and then we load them on a truck. And in Grand Falls, IRMA packages them up in a similar way and they put them on the Cross Island bus. And they send them to me across Newfoundland on the bus. We generally get them about 10 o'clock at night. And then we do experiments in the lab through that night. So it's the same day that the gametes have been collected. We do these elaborate split ejaculate and split brood designs in the lab where we manipulate all kinds of different things. We fertilize the eggs. We put them in recirculating flow through water in a temperature controlled room. We incubate them long enough that basically you can see the eyes, but this is for that they have enough tissue so that you can then genotype them. So if you take then a piece of their tissue, we already know who the mom is for each egg, and all we're trying to do is identify the species of the father. So whether the egg, if this is a salmon egg, whether it has a salmon father or a trout father, so whether it's a hybrid or not. So there's a fairly simple molecular test that is published related to this. So to confirm that it actually works, we purposely produced known hybrids in the lab. So not in sperm competition, where we took trout eggs, for example, and gave them trout sperm or salmon sperm, and we took salmon eggs and gave them salmon sperm or trout sperm, so that we have known pure individuals and known hybrids. And then we run this assay on them, and basically the double bars indicate the presence of a hybrid, and this works. Excuse me. So this is some of Connor's data. So again, this is Newfoundland fish. And what we have here is 15 blocks in a complicated design representing gametes from 60 different adults. If we look at the brown trout, and so these are Newfoundland brown trout that were brought from Europe starting in 1883. And these female brown trout, just like their cousins, long-lost cousins in Europe, they do exert conspecific sperm preference against male salmon. So this is relative paternity. So if, a given group of eggs from a female, what proportion are fertilized by a trout father, which is in brown trout, and what proportion is fertilized by a salmon father in gray. And in this case, a salmon father makes it a hybrid. So we can see that the previous work I showed you is that the gametes are fully compatible, they hatch fine, but under sperm competition, most of the trout eggs are fertilized by a trout father, although there is some hybridization from salmon. When we look at salmon from the exploits river system, we see more or less the same thing, but it's not as pronounced. So whether these two bars are statistically different or not is not that important to the story here. But if we just look at the means, salmon eggs are predominantly fertilized by salmon males, but a pretty high proportion do get hybridized by trout. And so female brown trout exert conspecific sperm preference against male salmon, and the female salmon seem to have less ability to do this against male trout. And this fits our predictions going into this about what we thought might happen. Also, like in the work from Yates in Europe, published in 2013, the ovarian fluid that's released with the eggs seems to be really important in enabling this to happen. So this is a subset of the same fish. So this is 9 experimental blocks representing 36 adults. If we look first over here on the trout on the right, so the far right group here, these are trout eggs with trout ovarian fluid. That's the natural situation. So relatively few hybrids are produced. So salmon hybridize some of the trout eggs, but not a lot. But if you take the trout ovarian fluid away and you replace it with salmon ovarian fluid, then the proportion of the trout eggs that get hybridized goes up. If you go on the far left and you look at the salmon eggs and the salmon ovarian fluid, the natural situation, if there's almost 50-50 of the eggs in this group of fish were hybridized by trout sperm, but if you take the salmon ovarian fluid away and you replace it with trout ovarian fluid, the same sister eggs, the hybridization rate goes up. So if we switch these ovarian fluids, the hybridization goes up. And again, this is what Yates showed from Europe and we have from Newfoundland. So conspecific sperm preference exists and the ovarian fluid is important, but exactly what it does, I do not know. Finally, the last thing I'm going to talk about just for a minute or two here, again, is the next step here and connecting back our dots when we talked about the virgin and repeats. So ovarian fluid chemistry and physics does influence sperm. Consecutive repeat spawning salmon have different egg chemistry than virgins. I'm hypothesizing that repeat spawning salmon have different ovarian fluid chemistry than virgins. And therefore, for the same males under sperm competition, repeat spawning salmon have less ability than virgins to exert conspecific sperm preference against brown trout, and therefore hybrid fertilization rates are higher in repeat spawning salmon than they are in virgin spawning salmon. And so we have some progress on this. In 2017, we did an experiment for this. So synchronized gamete collection of salmon on the Exploits River and brown trout from Windsor Lake. At the time, I could see the eggs in mason jars, and based on the color of the eggs, I proceeded with the experiment in classifying the salmon as being a repeat or a virgin. The scales were taken from the salmon at the same time. They were sent to DFO Newfoundland, who independently interpreted the scales and got back their interpretations with me. And months later, we got confirmation that I was 100% correct in identifying the status of the female salmon. So the sperm competition experiments were between a male salmon and a male trout in a block design. So we did this with virgin eggs and virgin ovarian fluid, with repeat spawning eggs and repeat spawning ovarian fluid, and then switched the ovarian fluid. We repeated this with different blocks of fish over several weeks, incubated and preserved the embryos for paternity, and that's where we're at. So we're just about to start doing the molecular work on this. So I don't actually have any data on this yet, but we will have it soon. So I'm going to end there. I'm not going to go through this slide. I purposely had this here to leave up while I was doing questions. So this is just some other types of salmonid research projects that I have on the go that are not at all related to what I just talked about today. And they're up here for, you know, people see what's on the go if there's interest in collaborations or future seminars or something. So again, I'm just going to leave this on the screen while I stop now, and if we have time for some questions. Thank you very much.
Thank you so much, Craig. That was an excellent presentation. We will now open the floor for a couple of questions, and we may take a few and then ask you to email your questions either to Craig or to us here at the Foundation. If you have a question, please click the little hand icon on your screen to raise it, and I will be able to unmute you to ask a question. That's our preference, but if the function doesn't work, you can type in your question in the chat and I will read it out loud. So if anybody has a question, please go ahead. So Bill Haley has a question for you. I'm just going to unmute him. Bill, you're muted on your end. If you could please unmute your microphone. There we go.
Hi, Craig. Bill Haley from Marguerite Harbour here. I'm with Marguerite Salmon Association. Is there any reason to believe that your research would be different if you had it did the same thing in our watershed? We have an abundance of brown trout in our watershed. Some I've seen that have grown up to 12 pounds. And I think the province have also done some work similar to yours, trying to identify the percentage of hybridized fish we have. So is there any reason to think we'd be in a different situation?
No, I don't think so. I mean, there's probably some nuance, like I alluded to, with the level of sympatry, allopatry, like the exact, you know, history of the salmon populations vary a bit. Perhaps the history of where the brown trout came from vary a bit, but I don't really think so. But as I alluded to, Bill, I think you know, I'm trying to hammer home the point here, is that when people are, you know, quantifying how many juveniles are hybrids or something, what this can tell you is that, well, hybridization is happening, but it tells you nothing. I mean, it might tell you, I'll make up a number, whatever, like 8% of the juveniles are hybrids, but that tells you nothing about what percent of the eggs actually got hybridized and then didn't make it to that stage. And undoubtedly that's a higher number, but I suspect in some cases it's a much higher number. So the frequency of hybrid juveniles is a poor predictor about the frequency of hybrid matings.
The browns in our area here in Nova Scotia, the data that I've seen only goes back to introductions back to 1926, but there must be other data that would be similar to yours back to the 1800s, I would think.
There would be good data on it, as yes, and actually I'm collaborating with some people in Nova Scotia now on a bit of review on how brown trout in different ways might impact salmon, and I'm writing the part about hybridization, but others are writing different components. there would be records, and you know, and I don't think the context is really any different than it is here in Newfoundland. Like, I think this is has the potential to be an unrecognized problem.
Thank you very much. I enjoyed your presentation, Craig.
Thank you, Bill. I have one more question here, Craig, and then I think we'll wrap it up. Gerald Chaput said in, neat, how quickly does ovarian fluid get diluted when fish spawn in the wild?
Yes, so that's a good question, Gerard. So ovarian fluid might do several things. So we know how it affects sperm, but for example, the non-Newtonian nature of ovarian fluid, one of the reasons salmonids might produce so much, and we speculated on this in the paper we published on its physics, is that, for example, when it gets, the eggs are released into the water, the ovarian fluid, non-Newtonian nature, it's like a starch solution, like you push on it and hardens up. Might actually hold the eggs and sperm together for a second or two longer than they would and not get washed down the river quickly. So, you know, its evolution might have been for that reason. What matters is that, you know, most of fertilizations that are happening when the sperm and egg are released are happening within a couple seconds. Like, it's irrelevant what happens 15 seconds after the gametes are released. The sperm have been washed down the river, and regardless, the other bloke has fertilized the eggs because they're spawning in sperm competition. So it only really needs to be there for like literally a second or two to matter.
Okay. That seems to be the end of the questions so far. If you do have questions later, please feel free to reach out to Craig directly using his email, cfpurchase@mun.ca, or you can contact us here at the foundation and we'll pass the question along to him. We've recorded the session and it will be available on our YouTube page. I would like to remind everyone that our next webinar will be held on March 12th with Christine Stortini from DFO. The title of the webinar will be Freshwater Climate Risk Index, a case study for Atlantic salmon in Eastern Canada. So a big thank you again to Craig and to everyone who participated today. We will hopefully see you at the next webinar. Thank you.